So our next speaker is Kenneth from Yale University.
Okay. Hi, everyone. I'm Ken Hoehn, a postdoc in Steve Kleinstein's lab at Yale. And today I'm going to be talking about my work in developing new phylogenetic methods for understanding B cell migration, differentiation, and evolution over time. So a bit of a word on scope for this talk. I'm just going to be focusing on B cell migration and differentiation. But for my poster, I'm going to talk about all three. So if you're interested in evolution over time, that's going to be detailed in my poster, which is number 208. So the focus of this talk is B cell lineage trees, and B cell lineage trees are models of the series of mutations leading from the unmutated clonal ancestor of a B cell clone to each of the observed sequences at the tips. So the tips of these trees represent our observed BCRs, and the branch links represent somatic hypermutations. One of the most useful aspects of B cell lineage trees is that they provide a historical axis to understand different sources of B cell diversity. So if we have B cells from biopsies from different tissues that are present in the B cell lineage tree, then the tree can be used to understand connectivity between those tissues. If we have different B cell subtypes within the tree, then this can be used to understand cell type transition events. And if we have samples over time, then lineage trees can tell us about persistence or reactivation or evolution of different B cell clones over time. For this talk, I'm just going to be talking about these first two. And just to give you an idea of our goal, or perhaps I should say our dream, is to start with BCR sequences that are annotated by tissue or cell type or isotype, build our phylogenetic trees with the tips labeled by those different trait values, and then use the structure of those trees to infer patterns of migration and differentiation between these different cell types. So this is a complex problem, and it's quite common that in statistics, complex problems are initially investigated using simpler summary statistic-based approaches. So rather than jump into a fully fledged statistical model of B cell migration, we first developed a maximum parsimony-based approach. The way this works is we first estimate our lineage tree topology using whichever method we like, and we label the tips of that tree by our different B cell subtypes, although it could be different tissues or different isotypes, which are here shown in different colors. We can then use a maximum parsimony algorithm to predict the series of cell type transition events within the tree. So this algorithm finds the set of internal node labels that minimizes the number of transition events along the tree topology. So this would be an example of a predicted switch between subtype 2 and subtype 1. We can then compare the proportion of switches from each of the cell types in each direction in our observed tree to the types of switches that we observe when we shuffle the cell type labels within our tree. So this tree that I've just shown is the same tree topology, but we've randomized the cell type labels. And if we perform that permutation test enough, we can build a distribution which represents the difference in the proportion of cell type switches from subtype 2 to subtype 1, in this case, to the proportion of switches between those two cell subtypes in our permuted trees. So the positive bias of this distribution represents an enrichment of cell type switches in our observed tree compared to our permuted tree. And while I'm going to be showing a lot of these distributions, in practice, what we're really interested in is using this test to calculate p-values for this relationship. And the p-value is just the proportion of this distribution to the left of 0. So this test, which we call the SP test, can be used to identify biased ancestor-descendant relationships in trees. To make sure this method worked and to find situations where it didn't work, we performed simulation-based benchmarking under different situations of the overall rate of switching and the relative rate of switching between two different cell type locations, which we're just going to call A and B. On the top left, We have totally unbiased switching on a slow rate. So you can see there are cell type switches in either direction. Below that is unbiased switching, but at a fast rate. So also switches in either direction, but there are more of them. On the top right, we have biased switching. So all of the switches occur from A to B, and below that, we have biased switching at a fast rate. So to determine whether the SP test worked and under what parameter space it worked well, this is a representative example of our results. So on the left, we have our results from slow switching, and we can see that with biased migration, we have a very clear positive bias in this distribution, which is what we would expect from the method. And if we have unbiased switching, we have a zero-centered distribution. So this indicates that the method correctly differentiates biased and unbiased switching if the switching rate is not too fast. As the switching rate increases, the distribution of trait values at the tips is less distinguishable from random, so the power decreases. So the SP test can detect bias character changes along trees as long as the switching rate isn't too high. One of the most interesting applications that we found for this method is in understanding migration of B cells from the thymus to the blood in myasthenia gravis. So myasthenia gravis is an autoimmune disease caused by autoantibodies binding to the neuromuscular junction. A standard treatment is thymectomy, but remission can often take years. And one hypothesis for why this happens is that pathogenic B cells arise from the thymus, which is odd because B cells really shouldn't be in the thymus in the first place. And some of them escape into the blood and periphery where they can continue to cause disease. As this model would predict, when we remove the thymus in thymectomy, we remove the source of these pathogenic B cells, but the B cells that escaped remain in the periphery where they can continue to cause disease. To test this hypothesis, we obtained paired thymus and blood samples from 4 myasthenia gravis patients. We found a substantial amount of clonal overlap between the thymus and the blood, and this tree in the middle is a representative tree of those patients. So we can see a lot of sequences from the thymus as well as some blood samples near the more distant tips. And if we perform the SP test on this data, we can see a very clear bias from thymus to blood in 2 of these patients, which supports the model that these pathogenic B cells are arising in the thymus before escaping into the blood. And if true, suggests that myasthenia gravis could be better treated by supplementing thymectomy with peripheral B cell depletion therapies such as rituximab or early targeting of the thymus in disease. Another application is in understanding isotype switching. So isotype switching poses an interesting challenge because isotype switching can only occur in a particular direction. So in humans, it can only occur in the direction shown on this slide. However, if you label your tree tips by isotype and then perform standard parsimony reconstruction, you'll frequently get impossible switches within the tree. So this circle shows switching events from G2 to A1. And biologically, those can't happen. So we developed a constrained maximum parsimony algorithm that forbids switches that occur in impossible directions. And this allows us to more accurately reconstruct the series of switching events along the tree. We use this to investigate the class switching origins of IgE producing B cells. So IgE is the primary isotype in food allergies. And there are a couple of different models for how it arises. One is through direct switching from IgM straight to IgE. Another is through sequential switching through an intermediate isotype. And another is complex, where all of those occur. To test this, we looked at a previously published data set from Nielsen et al. In 2019. And we found a very clear association between IgE and IgA1 in one peanut-allergic patient. And this supports the sequential switching model from IgA in this subject. Now, this method is very useful, but there are some caveats to keep in mind. The one thing that we identified is that there can be a higher rate of false positives if the migration rate is very slow relative to the mutation rate. There can also be some skewing of the results by biased sampling of different tissues, as well as differences in mutation rates over time between cell types. There are workarounds for these. One of them is to use a downsampling algorithm, which we've implemented to control for very slow migration rates. You can also check if your results are correlated with sequencing depth between different tissues, and just carefully interpreting your results if you're mixing cell types that have very different mutation rates over time, like germinal center B cells or memory B cells. So we've implemented these tests.
2 minutes.
We've implemented these tests in a new R package as part of Immcantation called dowser, named, of course, for the practice of using a Y-shaped stick to make sometimes questionable geographic inferences. This allows you to very easily build trees as well as analyze discrete and continuous traits along B cell lineages. So everything I've talked about here and on the poster is in dowser, and it's compatible with everything else in Immcantation. So thank you for listening. If you're interested in dowser, this is the URL as well as the URL for Immcantation and my email. I'm at poster 208 if you would like a more in-depth discussion of these. I'd like to thank our funders over the years, as well as my supervisor, Steve Kleinstein, as well as my DPhil supervisor, Oliver Pybus, and our collaborators. If you're interested in the discrete trait analysis work that we've been doing, it recently came out in PLOS Computational Biology. And we also had a recent paper in eLife detailing methods for detecting B cell evolution over time. And with that, I can take questions.
Thanks, Ken. Questions?
Hi, that's great. So, I mean, one advantage to applying these phylogenetic models to the B cell repertoires is there's so much data. Can you imagine trying to estimate evolutionary rates in different parts of the trees as a, you know, it sounds like you're looking at mixture a little bit, but actually estimate the rates of evolution in different clades.
Yes, so the rate of evolution over time? Yeah. So we've done some work in understanding, basically, if you look at B cell lineages that are sampled at different time points, can we see a clear relationship between increasing divergence and sample time? So those are basically treating the whole tree as one unit. But one thing that we would really like to do would be to develop models that allow different parts of the tree to be evolving at different rates. But that's definitely an area for future development.
Question from Marcos Vieira. Would we have enough power with typical data sets to simultaneously look at two types of transitions? For instance, if certain isotype transitions are more common in certain tissues, or if migrations are more common for certain cell types?
So my experience from that is that bulk sequencing tends to give you enough sequences to be able to use these methods for detecting different types of cell type transitions. And we've, in some of the application papers that we've written about this, we've been able to look at different cell type transitions in different directions with multiple tissues. For single cell data, it may be a little hard to do that, if only because the throughput tends to be lower and it's harder to get the big clones that you need to estimate these parameters.
Okay, I see no more questions. So thanks again, Ken. And once again, his poster number is 208.